Hello, welcome to our Lincoln University and Missouri River Regional Library lecture about cholesterol and its cholesteric friends. I am Vianney Gimenez-Pinto. I'm an assistant professor of physics and chemistry at Lincoln University of Missouri, and I'm very happy to be here sharing this program with you. So normally, when we talk about cholesterol, we assign it a very negative connotation. We think about heart disease and heart attacks and risk for stroke. But is it really cholesterol such an evil compound? Is it really this monster that we think every time we hear the word cholesterol? Well, actually, cholesterol is just a molecule. It's this cool compound that we can see here, and this is the molecular diagram, the full drawing of the molecule. That is our cholesterol. It's an organic molecule. It's a biomolecule. It's present in our bodies. And there is something cool about this guy. It's a steroid. And when we think about steroids, we also think about creating muscles and bodybuilders. But in chemistry, a steroid is just a molecule that has 3 rings with 6 carbons that are represented here with hexagons, with bright yellow hexagons, and then 1 ring with 5 carbons, okay, which is this green pentagon. Every time you have 3 rings with 6 carbons and 1 ring with 5 carbons in this specific configuration, you have a steroid. So cholesterol is one steroid, is a steroid in our bodies, but it's not the only one. There are other particular steroid hormones that are in our bodies. Cholesterol is, however, the most abundant steroid, and it has a critical role in our bodies. People normally like to avoid cholesterol. They think cholesterol is this bad thing, but actually cholesterol is critical for life. Without cholesterol, we will not be alive. It is critical for animal life, including our lives, humans, if we think of humans as animals, and it's also critical for plant life. And the reason why cholesterol is so important is because it has a function in our cell membranes, both in plants and in animal cells. Here, we're seeing this diagram of the plasma membrane in an eukaryote cell, and these membranes are an assembly of several components, okay? So let's go one by one. First, we can think of this membrane as a bilayer made of some molecules called phospholipids. And there are those molecules here that they have these red circles, okay? And then they have something like a leg getting out of that circle, okay? So what occurs is that those phospholipids are assembling into layers. We have top layer here with our red circles pointing up, and then we also have the bottom layer here with our red circles pointing down, okay? And we can think of this cell membrane as a double layer, okay? Layer 1 and layer 2 of phospholipids. But, of course, plasma membrane is not just that. The plasma membrane also contains proteins. Here we have a glycoprotein. It's a protein with a carbohydrate attached. And you are also going to find in our plasma membrane cholesterol embedded in here. You see? So cholesterol is part of our plasma membrane, and it's very important for maintaining that membrane functional. Why is it so important to maintain that membrane functional? Well, let's try to simplify this, and we are going to just draw the phospholipids in this cartoon. And what we see here is that when you have low temperature, when you're chilling your membrane, those phospholipids are going to be organized and they're going to be tightly packed next to each other. Okay, so at low temperature, my membrane is very rigid. It's not fluid, okay? There is not a lot of mobility in these phospholipids. They do not have a lot of space to move around, okay? Because they are tightly arranged next to each other. So the distance between them is very small. They are tightly packed, and they are just making a rigid assembly, okay? So we have a rigid barrier for the cell, and that's not good. I mean, our cells are supposed to be flexible, right? So that's what happens when if we had only phospholipids in our cells, and we will not have any cholesterol. So that would be our cell membrane without cholesterol. Now, what happens when we have the cell membrane with cholesterol? What occurs is that cholesterol likes to insert itself between the phospholipids, okay? And then what occurs is that it separates them a little bit, not too much, but a little bit. And overall, then my phospholipids are not as tightly packed. They have some distance, the average distance between them, it's larger. And as a result, my membrane is not rigid. It's a fluid barrier, okay? So that allows molecules to pass into and out of the cell. What happens at high temperature? Well, at high temperature, what occurs is that my phospholipids are going to have a lot of energy. And if they have a lot of energy, what is going to occur is that they're going to be away from each other. They tend to spread out. And you can find them, with an average distance between each other that is large. So the membrane is spread open, and it looks like if we don't have a barrier anymore, okay? In here we had an assembly, in here we had an assembly, but then in here my phospholipids are by themselves. So it seems like our cells now do not have a barrier, they don't have a plasma membrane, and that doesn't work well. So what occurs is that cholesterol inserts between our phospholipids, and then at high temperature, this phospholipid is attracted to the cholesterol molecule, and this phospholipid is attracted to the cholesterol molecule as well. So as a result, my phospholipids are closer to each other thanks to the action of the cholesterol. So at high temperature, instead of having my phospholipids apart to each other, now my average distance between phospholipids is reduced, and my membrane is still serving as a barrier. So at high temperature, cholesterol stabilizes the membrane function. And at low temperature, cholesterol helps to increase the fluidity of the membrane. So, cholesterol is critical for life because it stabilizes the fluidity in the cell membrane. And this is valid for plants as well as for animals. It's critical for life forms in this planet, at least. So if we don't have cholesterol, then we will not be alive. Beyond this role in cell membranes, cholesterol is also critical for human life, for what it makes us human beings. What occurs is that cholesterol is the starting compound for other crucial biomolecules. Testosterone and estradiol, which are the sex hormones that are common in the male gender and the female gender, are actually cholesterol derivatives. And both of them are synthesized inside our body from cholesterol. Also, if you remember when we were talking about steroids, these two molecules have 3 6-carbon rings and the 1 5-carbon ring. That is a signature for steroids. So actually, sex hormones are steroids as well. And those steroid hormones are synthesized inside our body from cholesterol. So, if cholesterol is so cool and so great and so important for life, then what's the deal with cholesterol? Why do we have to get an annual checkup and make sure that our levels are within a healthy range? Why is it that we need to avoid junk food, okay? Avoid, reduce our intake of animal fat, and try to have a more whole diet, a more complete diet, including olive oil and beans and oats and sources of good cholesterol instead of sources of bad cholesterol, has these fatty fat, animal fats are shown in here. Well, we have to think about these common names of good cholesterol and bad cholesterol. And actually, both our good cholesterol and our bad cholesterol contain exactly the same cholesterol compound. It's exactly the same. Our fella here with his 3 6-carbon rings and the 1 5-carbon ring is the same whether it is in a good cholesterol or in a bad cholesterol. Then you will think, then what's the difference between good cholesterol and bad cholesterol? It's all cholesterol and it's all good. Well, not really. Here's the key. Cholesterol travels in the body via lipoproteins, and lipoproteins are this just spherical bubble, okay, that embeds cholesterol, actually embeds lipids. It's not just cholesterol traveling in lipoproteins, but we're talking about cholesterol, so let's just think that cholesterol is there inside our lipoproteins. And actually, I want to simplify the drawing of this lipoprotein, so I'm actually just gonna say that it's a circle, okay, it's a sphere that is containing my cholesterol, and that's gonna make my visualization much more easier. Okay, so there are two types of lipoproteins that can carry cholesterol. One of them are lipoproteins that have a small size, and those are the source of good cholesterol. Those are the high density lipoproteins, and those are what made our good cholesterol. These lipoproteins are around 5 nanometers in size. So we are talking about the nano domain when we're talking about these good cholesterol lipoproteins. So the cholesterol that is transported, that is traveling in these smaller high-density lipoproteins is normally carried through the liver to be disposed. But then there are other type of lipoproteins that are larger. The whole difference is in size. In this case, this lipoprotein should be around 20 nanometers. And this one is the source of our bad cholesterol. Larger, low-density lipoproteins are the bad cholesterol. So when we are checking our cholesterol levels, what we are checking is the amount of low-density lipoproteins containing cholesterol, okay, versus our high-density lipoproteins that also contain cholesterol. In general, we want the number of smaller lipoproteins to go up and the number of larger lipoproteins to go down. So it seems like cholesterol is good or bad not because of its chemical identity, but because on the type of transportation he decided to ride in. If my high density lipoproteins are smaller cars, then we want to have more smaller cars in our body than these large trucks in our body. The reason for that is because those large trucks, those large lipoproteins, actually tend to accumulate in our arteries. So when you have a high number or a high concentration of these low density lipoproteins, of those large lipoproteins that carry cholesterol, then those large lipoproteins can create a traffic jam and start accumulating plaque in our arteries. And that is when we get into the risk of having a heart attack, heart disease, and a variety of complications that we do not want to have. So in general, cholesterol, from my perspective, cholesterol is a compound, and it's just a compound. The way it travels in our body, it's the problem. If our cholesterol travels in smaller lipoproteins, if our cholesterol is traveling in a smaller car or in a smaller medium of transportation, then it's good for us because those smaller lipoproteins can move around, have higher mobility because they're smaller. However, if our cholesterol is traveling in a large medium of transportation, in a large lipoprotein, then there is a chance of getting a traffic jam and to have accumulation of plaque in our arteries. So that is my take on whether cholesterol is good or bad for us. It depends on how is our body cholesterol traveling. So how can we prevent this? Well, I am a physicist and a researcher, a material scientist, so I'm not qualified to offer medical advice for your bodies. So that's why I went and checked the recommendations for the Centers of Disease Control and Prevention, the CDC. And they have some very reasonable recommendations to keep your cholesterol in healthy levels. So we want to prevent high cholesterol. We want to avoid the levels of LDL, low density lipoprotein cholesterol. That's what LDL stands for, and it's a bad cholesterol. And one of those is, of course, achieving a healthy weight. For that, we need to make healthy eating choices, maintain a healthy weight, get regular physical activity, quit smoking, limit alcohol, and if for some reason you get diagnosed with high cholesterol levels and you get a prescription medication to lower your cholesterol levels, please take the medicine as prescribed and follow your physician recommendations. So that is my warm-hearted advice for all of you regarding to medicine and taking care of your bodies. Always follow the recommendation of the experts in medicine. So that is the idea of our cholesterol within our bodies. Cholesterol has been a really interesting molecule and has made some serious contribution to the development of science and technology. If I take cholesterol and combine it with benzoic acid, I can get a cholesterol derivative that is very interesting. And this one is cholesterol benzoate. Friedrich Ritzner was a chemist and a botanist, and he was performing experiments with cholesterol benzoate when he found this particular behavior, these two melting points. So this material was the first observed materials that had both liquid-like properties, which means it was disordered, and crystal-like properties, which means it has some order. When we think about a liquid and a solid, we have this idea that liquids flow, right? And then they are refreshing to drink and swim on them. And then a solid, here we have a view of an ice crystal. It's just water, but in its solid state, in its crystalline state. But then when we go and think about the states of matter, actually the liquid state is a state in which my molecules are loosely bounded to each other, but they're free to move. And they are arranged in a disorganized way. While the crystalline state has a very strict and periodic order. Okay? So the liquid state is disordered. My particles here are in all directions. in all positions. Okay, there is no order, no pattern that we can see here. And then in here we can see a pattern. We have these cubes, and then my particles are in each of the corners of my cube. Okay, that's the main difference at the configuration level between states, between liquid states and crystal state or the solid state. But then cholesteryl benzoate had a state somewhere in the middle. It has something of liquid and something of crystal. And that was outstanding. So in 1888, Otto Lehmann, armed with polarizing microscopy, investigated the properties of this material and coined the term liquid crystal. When we think about liquid crystals, we think about liquid crystal displays. And yes, exactly that. A cholesterol derivate was the first observed liquid crystal material. Yes, those are the liquid crystal materials that were able to change the industry of monitors and TVs. Now we do not have monitors or TVs using cathodic tube rays. So we do not longer have this old-fashioned displays. Now our computers and our TVs look something more sleek and elegant. We have these thin displays that allow us to have compact laptops and to carry our computers around. I cannot imagine how a laptop will function if we didn't have flat display technology. Beyond just our TVs and our monitors, we can also think about the displays in digital watches, okay, and chronometers, and also the displays that we have in our calculators. Those are all forms of liquid crystal cells and liquid crystal displays. But beyond these well-known applications that we can see in our everyday lives, there are also cutting-edge research on liquid crystal materials that is occurring right now. One of those applications is their recent implementation of liquid crystal materials to move bacteria into preset ways. So bacteria normally will move in disordered paths or disordered trajectories, but then liquid crystal can make bacteria move in specific patterns. And here we have an example of the bacteria traveling in circles due to the presence of liquid crystal material in this sample. Also, order from liquid crystalline materials can make rubber to adopt some interesting shapes when temperature changes. So those interesting shapes can go from coils to cones to saddles, and these wiggly structure that resembles an accordion. Beyond that, light-responsive liquid crystal materials combined with rubber can convert light into helical motion. So this is a sample in which you're combining a liquid crystal material with rubber. Think about a rubber band, but a rubber band made with rubber and liquid crystal materials. And when you shine light into a rubber band, the rubber band curls. So you're converting light into helical motion. Shining light makes this rubber band to coil. And beyond that, light-responsive liquid crystal materials combined with rubber can make a rubber band that walks. what is occurring here is that we have a light responsive liquid crystal material and they shining a light on the side. And so having that light on the side create an asymmetric motion. And that asymmetric motion is creating this forward walking that we see here. So this is a liquid crystal material that responds with light. And then when light shines on a side, the whole rubber band moves towards a side. So all of these mind-blowing technologies and new discoveries started with our homeboy cholesterol and its derivative, cholesterol benzoate. So cholesterol derivatives had a liquid crystal state. They had this intermediate state between a fluid, a liquid, and an ordered crystal. But it was a peculiar liquid crystal state. And in order to explain this, I'm going to do something that some chemists, my chemist friends, may get upset by this simplification, but this is something that I have to do in order to explain what it will be the configuration of molecules, okay? So when we were thinking about a liquid being a disordered set of dots, we were saying that each of our molecule was a dot. Actually, that's not very good to say because my molecule is elongated. So a better thing to say, which is still an approximation, and I hope my friends that do chemistry don't get very upset with me. Please forgive me for what I am about to do. We can say this molecule is our rod, okay? And this is something in order to be able to describe what our molecules are going to be doing in this liquid crystal state. On average, these elongated molecules arrange themselves following a spiral helix, similar to what we are seeing here. And the materials that follow this helical configuration are called cholesteric liquid crystals. So what occurs is that my molecules are rotating, okay, following a helix or a spiral. So in this part, my molecule is aligned completely horizontal, okay? However, in this part, my molecule is going into the screen. And that's why you see it shorter, because you're seeing the short side of the molecule. Here, this will be the side view of the helix in alignment with this beautiful picture of a helix that we have here. And this will be the top view of my molecules at different part of the helix. So when they are horizontal in here, so seen from the top, they are also horizontal. But when they are shorter in here, seen from the top, is not that they are shorter molecules, is that they are arranged diagonally, okay? And then when there is this short oval here, it doesn't mean that the molecule is a shorter oval. It means that, seen from the top, my molecules are vertically oriented. So they are pretty much rotating from horizontal to diagonal to vertical, okay? Then diagonal again, then horizontal, and such and such. So they're forming a helix. This helix in the cholesteric phase can be nicely oriented, and here we have a three-dimensional simulation of the cholesteric helix, nicely oriented. Or it can be disordered. And here we also have a three-dimensional simulation of the pattern of this disordered cholesteric helix. Here we have a beautiful picture of several disorder configurations of this helix, okay? And we can see all these beautiful patterns. We still have a helix. We still have a cholesteric phase, but it's just being distorted, bending around, and showing these beautiful patterns. And that is in contrast with this nice ordered helix that we have here. This nice ordered helix will be the same as this one, and in here we have these undulations and beautiful patterns that occur in our helix. These textures, disorder helix and order helix, are the base of the LCD writing tablets that we can find in the market. Finally, I hope I'm able to convince you that our cholesterol molecule is not such a bad guy. It's not such an evil compound. It has an important role for our lives, and it has also contributed to the development of some really mind-blowing technology devices. Still, it's important to keep our health in check and to keep our low density lipoprotein cholesterol in a healthy range, as well as our high density lipoprotein cholesterol in a healthy range. And with that, I open the round for the online discussion. Thank you.